30 years ago. Oh, well, we were teaching evolution then, I think. There are some basic truths in biology that help link us all together, and one of them is that in evolution we all derive from a common ancestor, from the primeval slime 3.9 billion years ago. And since then we have diverged depending on where we live and how we live. And basically the eukaryotes, of which we are one, in animals over there on the right, are related to bacteria, aquifex down the bottom left, but only very distantly. And of course we were far more closely related to things like chimpanzees and mice and rats. So the more basically the more similar we are as organisms, the more similar our DNA is. However, because we are all related on some level, there are some elements of DNA that are the same across all spheres of life. And on this basis we can do a lot of computation with our DNA and our databases to see where the similarities are between DNA from different organisms. So I'm going to tell you quite an old story that illustrates the value of not only keeping the data archived, but keeping it archived in a way that it can be computed on, i.e. it can be reused by others coming after you doing your own experiment. So this is quite a long time ago now. There were some researchers in the United States who were looking at human colon cancer, and they discovered a gene that was implicated in human colon cancer, and they sequenced it. And this is represented by this line query at the top, and it's repeated 3 times. And what they did is that they took this sequence and they submitted it to the sequence database using an algorithm called BLAST, which is a sequence similarity algorithm, which basically said, are there any sequences in the database that are similar to mine? And lo and behold, there was a sequence that was similar that came up in the database. That's the subject, which is the 2nd, 4th and 6th line. And what BLAST does, it aligns these sequences so that you can see the areas of similarity. And here, the similarity is indicated by a repeated letter in the middle line. So you can see they're not identical, but there is a lot of similarity between the 2 sequences. So when they got very excited about this, because this is an E. coli gene. So E. coli is very distantly related to humans, but on some level it's related. And when they looked at that record for the E. coli gene, in the metadata there was a paper associated with that gene, so they clicked on the link to the paper, and lo and behold, that paper describes the role of that gene in DNA repair. So immediately, by doing not very much work, and actually leveraging the work of others that has gone before, they found out that the gene that they'd isolated, associated with human colon cancer, likely had a mechanism that involved DNA repair. I.e., when it's mutated and it goes wrong, the DNA is not repaired properly, so cancer ensues. All the cell processes and cell division goes wrong, basically. So that was their hypothesis. And that was allowable from the... of course, they had to go back and prove this with experiments. They couldn't just believe that, but this is a very strong indicator. And give them real pointers to do more experiments. Now, this paper was published a while ago, 1993, in Cell. And here it is in UK PubMed Central, and I don't know if this is an enhanced publication or not, I'm not sure, but here's the abstract. There's a text mining been applied to the abstract here, so we've got our gene ontology has picked out terms mismatch repair and chromosome, diseases has picked out hereditary non-polyposis colon cancer, polyposis colon cancer, genes and proteins mut-S, and MSH2, that's the names of the genes, the human and E. coli genes, and so on. We've got some funding information at the bottom. This is all funded by the NIH, and various institutes of the NIH. And at the top we've got the value-added stuff that UK PMC has done in the tabs. We've got citation tabs. This shows you the number of articles and the articles citing this paper. Bioentity shows you the terms that we've mined from the full text, if they're available, and the databases we've linked to. Related articles is a natural language processing algorithm that matches abstracts that are similar. It's a service from PubMed. So the PubMed ID of this, the identifier of this article is at the top, 8252616, with some magic syntax that is secret, which we hope to make less secret in the future. You can say how many articles cite this one using that identifier. what makes it unique in our space is the suffix med. And here they are. There's actually 849 articles have cited this one in our information space. And actually 2 of them here, this little multicoloured flag, signifies that we've got that article in full text available. One's in PLOS ONE and one's in the Journal of Gynecological Oncology. So all these papers have cited this one since '93. So that's quite highly cited actually. 800 or so is pretty good. Most papers get cited less than 50 times. Actually, half of PubMed's only been cited 0 times. So it's a famous paper. Actually, I should say, if you use UK PMC, there's a time-cited sort order there now, so you can, if you're looking at any list of any interest, you can sort by time-cited. And actually, this is what Mark Walpole at the Wellcome Trust did when we implemented that. The first thing he did was look for articles funded by the Wellcome Trust and then sort them by time-cited, see which was the best, so to speak. So one of these papers that cited the original one actually went on to elucidate the structure